Within the human gut, there are many microbial organisms which help the host to digest food and break down nutrients that the body isn't able to. These microbes are what constitute the gut microbiome and have a large influence in the gut-brain axis. The gut has even been alluded to as a second brain, as it plays such a large role in the central nervous system. Recent studies have been undertaken to determine the extent of influence the gut microbiota can have on mental health, specifically its possible link to autism spectrum disorder, or ASD. As the gut microbiome is so vast, it has been difficult for researchers to identify specifically which microbes could be affecting ASD, though it seems that the microbe can only affect the expression of traits and not completely rid of a person of symptoms. Recent studies have investigated the link between the gut microbiota and its potential effect on ASD, analysing the gut-brain axis and interactions. There have been found to be links to lowered levels of 2 bacterial species and high levels of 6 other species in individuals affected with ASD. Specifically, it has been hypothesized that some Clostridium species may be linked with regressive autism due to their ability to disrupt the intestinal microbiota. Supporting this, Maturazan and others in 2011 found the presence of a Clostridium species, Clostridium perfringens, which was significantly higher in those individuals affected with autism compared to unaffected individuals, highlighting this link. Another study was done into the effect of Dissolvio vibrio species and autism, finding that those individuals affected with autism showed increased levels of the bacterium in stool samples when analysed in comparison to unaffected subjects. Therapies for ASD are limited, and currently there are no on-shelf antibiotics or probiotics targeted for ASD. At the moment, it is a process of elimination to see what microbiota and their correlating ratios apply at the right amount to become therapeutic. With current ASD drugs formulated from other scientific approaches, the only gut-brain related therapy is trialling diets that are tailored to other brain pathologies, non-scientifically proven to work. However, there are many studies trialling antibiotics and probiotics to see how they aid ASD. In 2000, the antibiotic vancomycin was tested and found to improve 8 out of 10 children with ASD. Deemed not as a useful therapy, it did open the door for this research. Following up with many studies learning about what microbiota influences ASD over the years, there is hope for a probiotic in the pipeline using Bacillus fragilis. This species in our gut was recently found to positively change the social behaviour of mouse maternal immune activation models that can be related to human ASD-like behaviours, with many of the following studies can prove this. Clinical trials are still yet to happen, but this could initiate the uprising of future probiotics, pre-birth biotic transplants, and tailored nutrition yet to be made for treating ASD. While for this research and many other studies alike show credible evidence for the relationship between the gut microbiome and autism, there are several key issues. Firstly, a large portion of current research appears to be correlational. It is hard to tell whether the results that are occurring are due to the initial variations in the gut biome causing autistic-like symptoms, or if participants with autism can experience significant differences in their gut biome due to the disorder. Next to this, many of the studies currently available display a large sample size, which limits the amount of inference that can be made. While mentioned research does show a use of a control group, with such small sample sizes, it is difficult to determine if these results are due to the variance caused by individual differences.